We've digitized the 3D coordinates of these landmarks. We usually digitize 6 correlates on the top of the colony and 6 on the side or the edge of the colony. And to do this digitizing, we use a reflex microscope, which is a stereo microscope adapted so that a small light spot appears in the field of view. Essentially, there's a motorized stage with which the user can move the light spot into the right position and then send the coordinates to the computer. Okay. So the next step involves processing the data, and usually we use Bookstein size and shape coordinates to process the data. In other words, we create lots of different ratios, and we use those ratios as variables in statistical analyses. And this particular statistical analysis was based on 30 colonies. So these are the 30 colonies. There are 10 of Fabulata. These are genetically characterized colonies. 10 of Franksi and 10 of Annularis. And what we've done is we've performed a big canonical discriminant analysis in which the colonies form the different groups. And we've determined the distances between the colonies and used those distances to perform this cluster analysis. And the cluster analysis shows that the 3 species are distinct, and also that the Monastery Annularis and Monastery of Franksi are morphologically more similar, which is similar to the molecular results. In terms of the characteristics that distinguish the 3 species, you can see that from these SEM images that it involves the relief of the septa. Fabulata has higher relief, shorter extensions of the septa. Franksi has lower relief, longer extensions to the septa, and Annularis is somewhere in between, but closer to Franksi. Okay, this is another analysis. I have lots and lots of discriminant analysis to show you with regard to species problem. And in this particular analysis, the polygons represent the species in Panama, and each one of the points represents a colony. And you can see that Annularis is down here, Franksi is there, they're overlapping a little bit, and Fabulata is over here. And these dotted polygons, those indicate the 3 species in the Bahamas. And from this you can see that they're overlapping an intermediary, which suggests hybridization is going on in the Bahamas in agreement with the molecular results that I showed earlier. So the big question is, could hybridization be occurring? And to address this question, my colleagues have performed a series of lab experiments in which they've crossed Fabulata and Annularis, and the resulting offspring, the larvae, are a double band. They're heterozygous, suggesting that they are hybrids. However, when you go out in the field and look for these hybrids, none can be found in Fabulata or Franksi. And many of the colonies, 10%, a little less than 10% of Annularis appear to be hybrids. But when you look at other loci, there's strong frequency differences. So my colleagues that are working on the molecular data think that hybridization is not occurring today. Okay, so this has led us to say, well, could it have been occurring in the geologic past? And to address this question, we have been going around the Caribbean to different islands and collecting Montastria on the terraces that occur along the islands. And here we are. This is the 125,000-year-old terrace. I think this is an Andros Island in the Bahamas. And we've been making collections along transect lines. And in the collections are organ pipe types of Montastria, columnar types of Montastria, mound-shaped types of Montastria. In other words, the same sorts of colony shapes that I showed you before underwater, but now, of course, we've got them in the fossil record, so they're a little bit mangled looking. Okay, so you can't work with colony surfaces when you work with these sorts of colonies. So instead of looking at colony surfaces, but we've been working with transverse thin sections. These are microscopic slides that have been cut through the colony as close as we can get to the surface. And then we've been digitizing the coordinates at this time of 27 landmarks. Again, 6 from the top and 6 from the side. That is, if we could get them, because sometimes it's hard to get all of them on fossil corals. And here's some of the results. The shaded polygons are the modern species in Panama. So this is Monastrea franksi, and this is Monastrea fabulata as it is in Panama today, and this is Monastrea annularis as it is in Panama today. And these other polygons are the 3 species in the fossil record of the Bahamas. And notice that once again, they are intermediary and overlapping, suggesting that hybridization was occurring back in the Pleistocene, not only today. Okay, so how wide is this hybridization? I'm going to show you results of other Caribbean islands to address that. First, I'll show you Dominican Republic, and this is the same terrace in the Pleistocene as the Bahamas. This is the 125 terrace in the Dominican Republic. And here there are 4 morphs, and there's an overlap between the organ pipe and the columns and the organ pipe and the massives, but there is not as much overlap as you see in the Bahamas, where it's really extensive. This is the modern collections from Belize, and here you can see the 3 different species with the X's are franksi, the dots are annularis, those being much more closely related than fabulata. And at the bottom here it says, the Pleistocene Dominican Republic and modern Belize morphotypes match the modern species in Panama, but they do not match the Bahamas Pleistocene morphotypes. Okay, so this is a map of the distribution of the complex, and this funny looking thing sticking out there, that's going to Bermuda, and we haven't sampled there. But anyway, I've shown you Bahamas, and there we were looking at the 125 terrace. I've shown you the south coast of the Dominican Republic, which again we were looking at the 125 terrace. I've showed you the modern of Belize and the modern of Panama. And right now the Bahamas is really different from everything else, and so we decided to pick another location that was on the margin of the distribution, and this next location is Barbados, which is down south. And Barbados is unusual because there are numerous terraces. The island is going up. And this is a map showing the various terraces. There's an 82,000-year-old terrace, a 105-year-old terrace, a 125,000-year-old terrace, a 200,000-year-old terrace, 320, 450, 640, and so on. So what we did is we sampled the 125, the 320, and the 640. And here are lots of discriminant plots. Here's Bahamas, just for reference. Here is Barbados at 125, so the same age as Bahamas. Here's older in Barbados, and there is more than 5,000-year-old terrace in Barbados. The modern species here are all in red, franksi, fabulata, annularis, and so on. And the new species, or the species that we found within each one of these terraces, are indicated in blue. In the old Barbados terrace, we found columns and plates and massives and platy massives and so on. And so over here are the summary of our results. The fossil morphotypes within each terrace are distinct. Yeah. Distances between fossil morphotypes are greater than between the modern species. You can kind of see that, but I'll show you that even better in the next slide. Only one of the fossil morphotypes within each terrace actually matches a modern species. Sometimes it looks like they match, but it's because I've only shown you a few of the canonical variants, and there are many of them to be taken into consideration. And then finally, when you throw all of these fossil morphotypes into one big analysis, the morphotypes in different terraces do not match each other. So each one of these morphotypes is different from the morphotypes that we found in the other terrace. So this suggests to us that speciation is occurring in Barbados. Lots of small speciation events. Okay. So the last kind of slide about this. Essentially the next step is we took all of our species and morphotypes and threw them into one big analysis. We had the 4 species from the Barbados 500 terrace, the 4 species from the Barbados 300, 4 from Barbados 125, 3 from Caymans 125, 4 from Dominican Republic 125, 3 from Florida 125, then the Bahamas 125, and then Belize and Panama recent. And we put them in one analysis, and we looked at the distances between the species. And what you can see from this is that these are distances between pairs of species within that terrace. And the distances tend to be much higher in Barbados than they do anywhere else, especially compared to the recent. Meanwhile, the distances in the Bahamas are much lower than anywhere else. So it suggests that something different is happening in these marginal locations. That is, the Bahamas, where we think there's hybridization going on, and Barbados, where we think there's speciation going on. And meanwhile, in these more central localities, not much is happening. So stasis in the center. These central localities tend to match the modern day situation. They're a little bit lower, but it's not statistically significant than the recent location. So we've got stasis in the center, and we've got unusual evolutionary events happening on the margins. Interestingly, current patterns flow through the Caribbean in this direction. So they start with Barbados, and then they move through the Caribbean, and then they end up in the Bahamas. And this ending up in the Bahamas during the Pleistocene would have been much more difficult than today, because the Bahamas were more isolated during the Pleistocene. Okay, so evolutionary novelty is concentrated at the edge of coral species distributions. Speciation occurs in source population, hybridization in sink populations, and the conservation implications. In addition to biodiversity hotspots, conservation efforts should focus on areas of high evolutionary potential. So both in terms of our analysis of overall clades within the Atlantic and also within species complexes, we find that there are certain areas where there's high evolutionary potential, and they do not necessarily correspond with biodiversity hotspots, so that in determining where marine reserves should be placed, these evolutionary hotspots should be taken into consideration. So, thank you.